Testing. 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 Testing.
So what we're going to do today is have a talk from Jean and Jiayu, and I guess you guys can organize yourselves. The topic will be data fusion. So I think you're going to do a bit of a survey, right, Jean?
Right, just introduce the variety of methods.
Yeah, great. Well, it's great to have everybody join. And sorry about 2 weeks ago with our WebEx issues, but I think those are solved now, hopefully going forward. So without further ado, we'll just jump right in and have Jean and Jiayu get us started, and then we'll have a little discussion afterwards.
Okay, I'm going to share my screen.
Okay.
You see my screen?
Yes. Yes, it's blank still, but hopefully, yep, now we see it. Press the project 2.
Okay. Okay, that's good. Yep. So the goal of this presentation, because we may plan to hopefully can finish this in 30 minutes. And we will cover several of the methods. And there's not the intention to give you all the detail of the message to see how it works. It's more of introduce you that there are this method existing and then as possible applications, then just try to stimulate your thinking about what possible method you can use for your own projects. And then for this presentation, it will be me and Jiayu, we're working together. So some slides I'm going to go over some slides, and Jiayu will take over some other slides. Okay. And so subject 2 is mainly focused on the multimodal data fusion. So first, I want to introduce what is data modality I use for.
Your sound is kind of a little cutting in and out, Jean. Maybe check it if you can. I don't know if there's anything.
I'm hearing an echo.
Are you there, Jean? Oh, she may not know that we can't hear her.
All right, we lost her, I think.
Oh, no, I think she must have.
Okay, I will try to.
Oh, okay. I can hear you now.
Okay.
Try sharing again. Let's see.
Yeah, let's try again. I think this time should be, because I was using VPN, so that's probably the reason.
Another thought would be if you email the slides to Dina or myself, we can put them up and have you just go audio only. But let's try this and see how it works for now.
Okay, can you hear me this time?
Yeah.
Yes, so far.
Okay, let's try this way. I can think that the VPN maybe slow it down.
Okay.
right? Okay, so back to the multimodal, and I want to first introduce what data modalities it could use for these projects. And brain imaging data, I assume everyone here is much more familiar, and the structural, functional, even the MRS. So we have a variety of data, brain imaging data can be used. And the second is brain signal data, which some may use, some have not yet, including the EEG and MEG. I know currently they have EEG projects going on, so hopefully some of the methods introduced here we can use for the EEG. And then there's a neuropsychological assessment or neurocognitive assessment that is mostly common for most of the other projects. Then the last set of genetic data is from genetics and epigenetics. It may not be very common, but that's particularly for me. I'm more interested in the combining the genetics, epigenetics, with the imaging and the other assessments. So overall, I bring this picture in. Then you can see, all this data we collected is from the very beginning of the genetics for the DNA codings and epigenetics, how to regulate the gene expression, and transcriptomics is directly related to the genes express itself. And then expressions become working to connect with peptides to become proteintomics is some of the data from protein. And after that, we have narrowed to the organs we're interested in is brain. Then we have the brain structural data and brain functional data. At the end, the highest level is the behavior assessments. So along the line, biologically, from very basics to very top behavior levels, all this data, theoretically, we all could combining them together, and then the information converged should provide better or more comprehensive power to understand the diseases, understand the biology, and understand how the brain and behavior functions. So that is broad. Yep.
Yeah, I just wanted to add one piece on top of this. This is great. Just as sort of an FYI, we've got another project. We're working on this sort of macro environmental project. And so you could even put something further to the right, which is we're looking at sort of satellite imagery. So measuring things like green space and light, city lights and water and things like that, and how those are linked to brain structure and brain function and et cetera. So we can go global as well. So
yes, thanks for the addition. That's right. That's exactly. We have the environmental piece added here. Yes, we have the study this micro in the environment in terms of individual environment, large to the area and the pollutions, everything can add into the environmental piece. Yes, thank you. So after introducing the data, now we're looking at, when we have this analyzing this multimodal data. So that is the focus of this subject is multimodal data analysis. We want to combine this information together. And then when we combine the information together, there's different goals, right? It's like different subjects, different projects have different goals. Therefore, the tools, the method used will be differently. So here is if the goal is discovered shared, linked information, shared information, or comprehensive, complementary. So when the goals differently, then the tools used will be different. Some is for the better understanding of biology or mechanisms. Some is for the better prediction, classification. Then this is a survey, this is a review done a while ago. I think it's Jingyu and then. So this is a list. So right now, this list of the tools can be used for the multimodal analysis. By no means, this is not complete the list. And recently, there's more data, more analysis of tools comes in. But just to give you an overview, saying how many methods can be used, a lot of methods can be used. And for our own group, our group as the trend centers, we can provide all this analysis. So for this talk, I only pick several of them. I will pick explain joint ICA and MCCA, multimodal canonical correlation analysis, or pick the parallel ICA and also our pick deep learning, which is additional ones. So the goal is give you a view of variety of the method can be used, and then each method has its own benefit, have own limitation, but it's only whether it fit into the particular projects. So now we go to the detail of each method. First is the joint ICA approach. This is Vince proposed the method early on. Then this method is to looking at the two modality. For example, here is a functional and here is a gray matter. And then for these two data modalities, the idea is if there is the component, for example, here is a component in this particular locations, and then there is one component in gray matter component in this particular location, they are coherently, they are variation, they are coherently varied across subjects. So for this strong linked component, if we concatenate data together and apply the joint ICA, then this is a simulation you can say that this method can clearly recover this linked component. And then you can see the variation across subjects can recover as the ground truth. So this is the theory of, basically the basic idea of this joint ICA is there are very strong connections between these two modalities. There are absolutely shared variation patterns, and we want to use to pull it out. So one applications here is apply this joint ICA approach to the functional MA data and EEG data, and these two are doing the same task. This example is to doing the auditory outbound task. So assumption is, okay, when this subject is doing this task, doing the same task, even though it's not at the same time, but their activation pattern should be very highly related if they do the same task. So based on that assumptions that we applied this joint ICA approach. So you can say that activations in the bilateral inferior frontal and temporal regions are linked to this P3 and N2 components. So these are basically is this activation in the location, activation pattern in the temporal profile are shared. Their variation pattern across subject is absolutely shared. So they can think about that it is absolutely joint component. Then by this information, we can get their spatial location. Now we get their temporal location, temporal profiles. And for this one, we also saw these temporal profiles have significant differences between patient with schizophrenia and healthy controls. And activation of are different. So patient has reduced activations as showed up in the both EEG and the functional MRI activation. So that is one typical application of joint ICA approach. And now I will switch to Jiayu, will go over the CCA, the CCA stands for canonical component analysis, and then she will introduce the CCA-based set of algorithm.
Thanks, Jing. Hi, can you hear me?
Yes.
Okay. Do you want me, maybe I can share my own slides?
Okay.
Oh, sure.
Okay, thanks. Okay, it's here. Can you see my slide?
It's coming up. Yes, we can.
Okay. Oops. Okay, yeah,
Yep.
they're here.
Mm-hmm.
Okay, so thanks for Jing for the introduction. So I will start here from the canonical correlation analysis. It's just like a data fusion method trying to maximize the correlation between two data sets X and Y by projecting them to like a different space. Like as you can see on this left side, you have linear weights A and B. Then you apply them to x and y respectively, you will get new vectors u and v. These new vectors will be ultimately aligned to get like the maximized correlations. So it's pretty straightforward. While this original version CCA is for two modalities, we also have the multimodal CCA, which can be used for like more than two modalities, 3 or 4. For MCCA, like one common used approach is just to maximize the mean of the sum of the pairwise correlations across all these modalities. But one issue for both CCA and MCCA is that they will likely overfit when you have high dimensional data, which is our case for our like imaging data. Usually it's high dimensional. So to address this issue, oops, why it's not moving? Okay. To address this issue, Jing Sui, she developed this MCCA plus joint ICA method. So as indicated by its name, it's just like you apply MCCA first, then it's followed by joint ICA. So you get these latent variables from MCCA, then it also has their like associated components. You can concatenate these associated components and then apply joint IC to them to maximize the independence across these joint components. That is kind of like to add additional constraint of independence. So your data will not have like maximized correlations. They were also like required to be like show maximal independence across these joint components. So this additional constraint helps address this overfitting issue. So this slide shows one application of MCCA plus joint ICA to 3 imaging modalities. So we have fractional and isotropic from diffusion data, then gray matter density from the structure data, and also ALF, the amplitude of low frequency fluctuations from the resting data. So after this MCC plus joint ICA, they identified several pairs of joint components. Then they tested for each joint component whether it's like underlying the 3 unimodal components showed group differences between schizophrenia patients and healthy controls, and this figure summarizes the results. So they have some like joint or like modality common discriminating components, so highlighted in these green frames, and the red frames are for those components which are just unimodally discriminating between case and controls. Particularly, the paper highlighted this large component, this joint component IC7, highlighted by this green frame. So it's a joint component where all its like 3 unimodal components showed group differences between patients and controls. It's like as mentioned in the paper, these like patterns, you would not be able to identify these patterns if you only do the in like unimodal analysis for individual modalities. It was only like identified by this joint analysis. So that's like one unique part of this approach. So it will give you some biomarkers with meaningful like clinical relevance. In addition, it tells you some like relationships across these modalities, give you some like joint brain network, and this like patterns and relationships, either like relationships is like only for joint analysis, but in addition to that, the network patterns is only it will only be extracted by this joint analysis, not available when you do individual analysis. So that's how you like want to use MCC and joint IC. That's the unique thing of this approach. So next I will move on to another group of methods, which is based on parallel ICA. So parallel ICA, the main difference between parallel ICA and those CCA-based approaches is that parallel ICA like prioritize more like the independence part rather than the correlation part. So in parallel ICA, you first do ICA separately on the 2 data modalities to make sure you have independent components. Then you try to maximize the correlation between the components associated loadings. So And here it is that you can choose how many component pairs you want to optimize. So given like you have several components for each modality and you have all these like combinations of component pairs, you can choose to like optimize one or two or three pairs. Like the rationale is that if like you expect your data just like modestly related, associated, you probably only want to have one component pair optimized. Then I feel like it's they are more like strongly correlated. You probably want to increase this number. But it should be noted that, like when you choose to optimize one component pair, it's not saying that you will only get one. It's just like a prior knowledge, and you tell the algorithm to optimize one component pair, and it is expected to help guide the learning process and to help to avoid or like reduce the possibility of the learning process to be trapped in the local optimum. So that's like a kind of a semi-blind approach. And so as it is designed, this approach is like maybe more suitable for data fusion, where the two modalities are like just like not very strongly related. For example, the genetic and imaging modality. So it's not like, as Jim mentioned, like those for joint IC, when you have two imaging modalities, they can be very strongly linked. But for genetics, it's like usually modest effect sizes. Like for example, you only have like you have to have like hundreds of thousand samples to identify significant SNP, which are related to some specific feature. So you would not expect very strong correlation between genetic and imaging modalities, and that's like where the parallelizing may find its like applications. So in this slide, we show one application of parallelized CA for data fusion between 1400 schizophrenia risk SNPs and 400K gray matter voxels. So as you can see, it was able to identify one linked component pair from 777 discovery samples. So on left side is just imaging component highlighting the inferior parietal regions. On the right side, it shows like a good difference between schizophrenia and controls. On this right side is the SNP component, highlighting the chromosome 6p22. This chromosome region, it's like one of the chromosome regions that show the most significant schizophrenia relevance in the large-scale GWAS studies. So it identified this component pair in the discovery, and it was able to show that these components and their associations replicated in another 600 new replication samples. So this like to prove that this like identified linkage is very reliable. So considering that you have like for GWAS studies, you need to have hundreds of thousands of samples to identify like reliable SNP association with features. Now with parallelized CA, it's like only less than 1,000 discovery samples, it was able to identify some reliable component and association, which can be generalized to new data. So it's kind of much like improved statistical power. However, so parallelized as it is designed originally, it's still not like it's not able to like handle like genomic SNPs, which are usually like between like 500K to 1 million. Why we want to analyze genomic SNPs, not just those like schizophrenia risk SNPs? Because like for schizophrenia risk SNPs, they were reported in the GWAS studies and they have to reach very significant p-value like 10 to minus 8 to be marked as reliably like relevant. But we still have some like suggestive risk SNPs. They may have some p-value like 10 to minus 4 level. They show some relevance, but not as strong as those genome-wide significant ones. But you cannot say that they are not important. And another thing is that these SNPs, they may interact with each other they may interact with those genome-wide significant more risk ones and to form like a genetic network to contribute simultaneously to the trait of interest. So you want to see how like these like top ones interact with the suggestive risk ones, and maybe how they interact with each other to contribute to their schizophrenia risk or other trait of interest. So that's why we sometimes we want to analyze the genomic data. So that's why we have this referenced parallel ICA approach. So the idea here is that you will provide parallel ICA with some reference names which you know are relevant to the trait of interest. Then the parallel ICA is instructed to make sure that these reference names they will have like they will be important. They will have very like they will be highlighted in the resulting component. And meanwhile, this prior knowledge is like again is expected to help guide the learning process, to have like more like global optimal. And so it's not like you will have these reference names highlighted. It's expected that these reference names will help elicit other SNPs interacting with them to be also important in the resulting component. So it's also kind of semi-blind approach. And we applied this approach to this data set is genomic SNPs, about 600K SNPs, and for the fusion with, again, 400K gray matter voxels. And we had a different linked pair here. On the left side, it's the frontal temporal component, which has been consistently identified to have schizophrenia versus control difference in different cohorts. And on the right side, you can see we have this Manhattan plot for a genomic SNP component, where we had not only those like reference SNPs, but also other like identified SNPs. And we show that this SNP component, it generalized to new schizophrenia data set, where this component also showed schizophrenia versus control difference in this new data set. So it's like reliable, like just prove that this newly identified SNPs together with the reference ones, their combined effects does show schizophrenia relevance. So this is reference parallel ICA, and besides, we also have some additional methods, like variations of parallel ICA. So first is parallel group ICA. It's like in brief, it's for unsymmetric data. So on one side, you can have 2D. On the other side, you may have 3D, like fMRI data, with the time sequence incorporated. The concept is that we can build a like similarity metric, which is the measure of similarity between the group level components and the individual level components. And this feature can be used for the fusion with the 2D data set. So that's parallel group ICA. And we also have N-way parallel ICA and anyway ICA. So those two are for data fusion where 3 or more modalities are involved. The only difference is that the N-way parallel ICA, it just tries to like maximize the pairwise correlations just one by one, like in a sequential manner. But for anyway ICA, it just like maximize the mutual information within the joint components. So that's two approach for data fusion with more than 3 modalities. And I think that's all for parallel IC-based approaches. Then next, this slide we have we briefly introduced the imaging transcriptomic analysis, which is mainly to just provide another perspective of data fusion. So it's not like all those ICA or CCA-based approach where we assess correlations between features along the subject dimension. So the features, like their correlation, are assessed based on their variation across subjects. But this may not be achievable when, like for the transcriptomic data. Like transcriptomic data, its gene expression is tissue specific, which means that if you want to have like the gene expression in the brain, you have to have brain tissue. That is like not easy to get. I mean, so like given this like limitation, so people are trying to do this in like with different strategies. So one commonly used approach now is that they link the imaging and the transcriptomic features based on spatial variability. So as you can see on this left side, so we have this like maybe a specific brain atlas. Then for each part of brain region, you can have one like you can have a transcriptomic feature, for example, the expression of a specific gene. And in parallel, you can have a like feature based on the imaging modalities. For example, the group difference between schizophrenia patients and controls in terms of their brain matter density in this particular brain region. So this like difference can be quantified by cohorts D, which interpreted as like a measure of this like this brain region's vulnerability to schizophrenia. And you do this for all the brain regions from 1 to n, and you get these two vectors. Then you can assess their correlations based on their spatial variability across these brain regions. So we also tried this on our own data set, and this table summarizes some preliminary results. As you can see here, we identified a couple of genes whose expressions are strongly correlated with the patterns of like case control difference of gray matter features in both BSNP and COBRA data. And particularly highlighted is this gene CALB1, because this gene is a pyramidal cell gene marker, which means that this gene is preferentially expressed in pyramidal cell. And pyramidal cell, we know that it's a type of cell which like are preferentially disrupted by schizophrenia risk genes. So this identified gene is consistent with the literature. So that's like how you can draw inference between based on the imaging and transcriptomic feature based on this type of data fusion. Okay, so I'll stop here, and next Jean will continue discussing some like deep learning approach for multimodal data fusion. Okay, Jean.
Okay, I can just use your slides. That's
Okay.
fine.
Okay.
Yeah. So maybe I just maybe add a little bit on Jiayu's the genetic portion. So we also did some analysis regarding DNA methylation, how DNA methylation is related to the genetic codings, and we also did the methylation with the brains and behaviors. So there's a lot of the data modalities were being used, and then we are right now focused on the data fusion method, right? How to have the method fuse the data from different modalities, but each data modality itself has some specific preprocessing which are different. How you pre-process the imaging data, how you pre-process the methylation data, or how you pre-process genetic data are very different. And our group, all have this experience to handling different variety of data. So then we move to this new model. This is the one which we probably many of the computer science backgrounds know deep learning approach to analyzing large data to get the complex features out and to classify or predict or regress some complex measures. This is a very upcoming complex models. And we also use this for some of our analysis. Here is one of the example that when we think about deep learning, it can apply to the single modality, but also can apply to the multimodal. When we apply to the multimodal, I want to say the first is you can choose different neural network frameworks in addition to how you fuse the two model. So first, in the neural network, they have different. This is only like 4 of typical neural network models. So the first one is the early ones is the deep belief neural networks, and then this is commonly used for imaging processing, convolutional neural networks, and here is recursive and more recurrent neural networks more related to the time series, and this one is autoencoder decoder networks. So when we try to using these deep neural networks to model things, we have to choose which basic frames we want to use. When we have that, the next one is how we build the fusion of 2 or multiple modalities. We can have a different way of fusing, but that all depends on the particular projects, what we're thinking about, what type of modalities we're using. For example, we could have a shallow future fusion, which is directly put all the data together, right? That can maybe it's maybe good if you have green matter volumes, you have white matter volumes, or the white matter tracks, which are featured, you can feature them together. We are relatively in the same level, and then combine the feature together. Then we're using the neural network to fuse the information from two data modalities, and at the end to do either classification or regression models. And then the other one is the deep feature fusions. That what the idea for that is when we have each modalities, we will do some modality specific analysis. We can that usually if we have the fusions from structural MRI or the fusion from EEG, then you may want to have some feature specific like domain or data modality specific features extracted first. Then when we have the higher level, you can combine them. Then you can think about, okay, we get the feature from EEG, we get the feature from structural MRIs, and then when we combine them because they representing the same brain regions, then we can combine them together. So that's how deep feature fusions. And when the feature combined, then we can do at the end do the classification or regression model. And the last way of fusion is the decision fusion. So we can train the data modality separately. We get the data modality specific features and data domain specific classifications or regressions. And then at the end, when the decision level, we combine them. So there are different ways to fuse the data. And we have using the deep learning neural network for fusing the imaging and genetics. And we usually is in this level. Like we have the features domain first, feature specific first, then combine them, then do the last classification or regression. Can we move next one? So this is one example published from the trends that is to the multimodal deep learning based for the structural, functional, and SNP data to do the classification problems here. So for this one, you can say the one modality is the functional MRI. We first did the domain-specific feature extraction, then we built the feature matrix. Then after the next matrix, that's a neural network to get the more reduced or more higher level abstract features come out. And this is the structural MRIs. We first get the brain components out, and then each brain regions, the brain matter density is the feature input. Then we use encoder models to find the feature representations. Then that's the SNP, and this is we based on the GWAS result to select the top or promising SNPs. And then we run based on the sequence input, then build the attention model. This is another data series, recurrent models. Then we get all the features together fusion. This one, and then we do the last model training is do the classification and problems. So we can see by these models that could have, when we combine the three data modality fusion together, the accuracy is improved. But when we do this imaging and genetic features to do imaging and genetic combined feature to do bring classification problems, we find out is most of the classification power is more come from the imaging data, and genetics only contribute a very small amount of information to do for the classification, which in certain senses makes sense because the distance from genetic codings to the behavior manifestation of symptoms is a little bit far. We have so many biological mechanisms going on. So for including genetics, I think the most benefit is come from understanding the genetic pathways from the genes, how the risk variations manifest in the different levels in terms of epigenetic levels, transcriptional levels, or tissue protein structure. So if we're understanding this biological representation of the risk, that will be the benefit to understand the disease's mechanism. So that is the future or the future directions our group want to do is we want to build some biological informed networks, and not only for purpose of classification accuracies, but more of understanding the pattern, how this risks is go through these different biological pathways to influence behaviors. Can we move next slide? Okay, so I mean, by no means that is not to fully complete this method. I know Roger's here. Roger's also have a set of tools is using the subspace analysis. So overall, in one word, like we have a lot of expertise and we built up a set of tools to do the data fusion. Hopefully this talk is give you some ideas for your own projects that you think about some of our tools can help you. So I will open up for any questions.
Can I talk? Yeah. Okay. Thanks, Jean. So yeah, so there's a lot of data fusion approaches and tools, and there's a lot of expertise here. And so hopefully we can just help people learn about what's available. And if I think for this CREST project, some folks will be developing new tools. Other people will want to use existing tools to answer scientific questions. And so there's a lot of, I think, possibilities there. So, but I also know that there is a lot of, there's a lot to sort of learn. So I want to sort of open it up for questions for people that we just have even just any questions at all. Basic questions like what is data fusion are good as well, or what is, how do you do correlation? Anything is gain here. So thanks, Jean and Jiayu. So let's open it up for questions, discussions, thoughts. And if maybe you could stop sharing. Sorry, one more thing. If you could stop sharing the screen, and then if folks want to turn on their cameras, that would be great. So we can kind of see you. Although you can't see us in the room, I know, so we're kind of unfair, but that's okay. So we're doing the best we can here. Thanks, Flor.
Sorry, do you want me to leave the slides open?
No, I would say turn off the sharing so we can see a larger.
Okay.
Yeah. So, did someone have a question? I cut somebody off.
Yeah, I had a question.
Go for it.
Basically, what you presented here seems very interesting, very exciting stuff. Very look forward to learning more about it. For definitely. My question, I guess, is on the more pessimistic side. Looking at I think by understanding more about the limitations of the various methods presented here, we would be able to have a more richer understanding of when and how to appropriately use said tools in various projects and research. So I think it's important to be excited and innovative, but it's also important to take a step back and look at where some of the limitations are and because by understanding that we can improve further, I think. So I think maybe if you could touch upon that more would be wonderful.
There are no limitations. No, I'm sorry. Just go ahead, Jean.
Yeah, that's a very good question. That's things you're really thinking about applying them. Yes, every method we have a limitation because other methods have assumptions, right? You're assuming something. When you violate your assumption, then their method is basically false. So you can think about, for example, joint ICA and CCA. Joint ICA assumptions, you have to think about the two modalities share exactly same pattern. If that fits your assumption, you're assuming that, then you can apply that. And for the CCA is purely looking at try to maximize the correlation of the two data modalities. When you have many of these variables, CCA based on the data very likely give you the false positives, overestimate the correlations. To count back that is you have to have different data to replications or to do more, either is limited of input of the variables or you have a second data to replication test. And then, so that is a problem with CCA. And for parallel ICA, like I mentioned, again, parallel ICA also have limitation of how many variables you input, right? If your variables is so big, and then as a more likely is the component you're looking for, you cannot find it because it's embedded by the large variance. So you want to have selections. Otherwise, you like Jiayu mentioned, you have the reference. You tell the algorithm which part the variance you're looking for is most likely come from where. So to help the algorithm to find that. Otherwise, smaller variance, you couldn't find it. So that's the limitation of that. And
I was going to say also in all of the sort of the primary papers on these different approaches, there are usually simulations and validations and things like that that will usually show where the method will break and where it won't break, right, in certain contexts. And so one example of that is parallel ICA. I think Jean had initially developed that, and she showed that, like, the sample size had to be a certain number before you could start to pick up robust relationships. So she kind of developed this little rule of thumb for sort of the sample size versus the number of genetic sites that you're looking at as a ratio, as a criteria for that. So yeah, and there's a ton of those things. So I think as the review paper, I would say that that Jean referred to actually had also briefly discusses some of the limitations and strengths of them. But yeah, very important question.
All right, thank you. Thank you so much for answering this question. Appreciate it.
Sure. If you have another question, just feel free to unmute and speak up, because we can't see all of your pictures on this. Oh, I think we will. Hopefully, yeah. See, we can't.
You don't do whoever talking.
Yeah. Did anybody just like zone out and say, "What the heck is all this stuff? I don't understand any of it," and want to know more? Raise your hand. No. So we're happy to work with people if you have. I think a good way to get started would be, because I think most studies today collect more than one modality, right? I think I would say all of them. And so if you have some questions that you're interested in trying to answer, and you're wondering, can one of these methods help me answer that question in the context of combining modalities, then just reach out to one of us, and we will we'll discuss with you and try to come up with some thoughts on, well, here, like as Jean had mentioned, in the case of parallel ICA, if you're looking at relationships between very different modalities with different effect sizes, dramatically different effect sizes, then this looser criteria in parallel ICA might be the best approach to start with. So, and then usually I think the way people learn is you don't learn everything at once. You say, well, here's my question, and maybe we say, well, this is a good one to start with, and then we get started with that. You try it out, and you see how it looks, and see how it works, and learn to understand it a little bit. And so it's not so overwhelming. You don't have to kind of learn everything. You can just start it with one little corner. Does anybody have other ideas about data fusion, or any comments on what else we might do? What else we might, what's the kind of question that you might have that you didn't see addressed today that we could, think about? Including those in the room here.
We just mix the data.
So Sergey's coming at it from we can learn anything given enough data perspective. So he just says, throw all the data in and it will magically come out.
The keyword is magic.
Magic. So yeah, so I think deep learning is definitely a growth area in the context of different many different kinds of data fusion. And, with deep learning, you can, in theory, universally approximate anything, right? And then learn all of the relationships from the model fit. So we will probably get there at some point, right? But
No, I don't think so. I think I see a different thing emerging, and it goes in waves, but the waves, nothing is new under the sun. We're saying it.
And all models are wrong, some of these.
Yeah. But there is another wave that's been there and that's kind of now coming back. It's differential programming, like treating deep learning correctly as differential programming. I think if we can substitute the parts of our models which we currently set by hand, like by hand, and learn them from data because we don't know how to program them, then that's where we will get the most out of deep learning. It's the places where we have data, but we don't know how to program decision widgets. But other pieces are working already quite well. We leave them as is. I don't know. Anyone with differential equation models?
So you're talking about like places where it requires a manual?
Yeah,
Okay.
current places where they have to choose starting point, boundary conditions manually. We don't even notice that we already used to the pipeline, but it takes a lot of hiccup, a lot of time waiting on us, on humans. So those places can be replaced now and automated. The intuition part of humans can be learned. It's not that what you said, everything can be replaced. There is no need to, especially for things that work. It's just if you have a logic system, this logic system can get other tools. Deep learning will be used by the logic system as an instrument inside of that pipeline. That's what I'm arguing.
And I would say it depends on the problem that you're trying to address too, right? In terms of how
Everything should start from the problem. I even disagree with your advertisement to the newcomers about try a problem. Well, Yoda said there is no try, you do it or not.
Yeah.
So you have to pick up a problem. Forget about methods, deep learning or anything, and work on this question and find the solution. It doesn't matter what that solution.
If you come up with a problem first, that helps narrow down what the options are, right?
That makes options.
Otherwise, how do you start? Like, how would you start? Otherwise,
Yeah.
You have to start. You have to get in this cycle, and also I think if someone's coming at it from the perspective of I've never done data fusion, I don't even know what data fusion really is, other than it combines two things, right? Then how do you get started, right? So I think getting something sort of straightforward, relatively straightforward, or getting a place to start, right? Start playing with some data.
Start.
Yeah. So that's kind of what I was thinking up there. Great. Other comments, thoughts, anything on data fusion? We have, let me just. Okay, so next week we've got Vanetta, right? So, and you're going to share some of your work on, sort of linking cognition and mood and looking at different types of data. Yeah. So.
It'll be more of a conceptual talk with some ideas about how to use some of these methods.
Yeah, that's great. That's great. We're looking forward to it. And we'll just kind of be, hopefully you'll be able to join us right at the beginning, but if you're a couple minutes late, because I know you had something right before, then we'll just have a little discussion and we'll wait for you to join us. So
Sounds good.
in the room, we'll get some bagels or something, right? Bagels and cheese.
Like what first?
So, okay. Well, thanks everyone for joining. We are at 2:54, so we'll let everybody go about 5 minutes early so you can, use that time and enjoy it while you've got it. And we'll see you all next week at 2 p.m. sharp for the next CREST research meeting. Bye. And for the rest of us on the leadership team, maybe we'll just stay on here for now instead of jumping to the other link. Or wait, you know what? I bet Kyle is gonna go, maybe we will jump to the other link, because I bet Kyle's gonna go to the other link.
Okay.
So, let's jump off this and go to the link that we sent you. You should have an invite for a 3 p.m. meeting. Can they join now?
I believe so. Let me just,
Okay.
I'm gonna close this one out. And
Okay, so we're gonna close this out and jump to that other link. We'll see you all in the other room.